You bought bread yesterday. This morning a spot. Green, black, pink, sometimes red. The fuzzy patches multiply while you sleep. Most people toss the loaf and move on, but behind those colors sits an entire kingdom of microscopic hunters, each one engineered by millions of years to colonize your kitchen. Some transform grain into medicine. Others pump out toxins so deadly they killed 100,000 people in 4 years. Bread mold is not one thing. It is a battlefield, and you're standing in the war zone. The first droplet of moisture, the first crack in the crust, and germination fires. Rhizopus stolonifer moves fastest. Aspergillus flavus comes next. Penicillium expansum thrives in cold. Cladosporium herbarum colonizes refrigerators. Neurospora setophylla turns orange. Fusarium graminearum stains pink. Every species has a signature, a temperature range, a toxin profile, and a body count. If you value documented mycology and food science history like this, subscribe. It is the best way to support our work. Also comment from which city you are hearing this from. This script is researched and factual, using sources from Science Direct's Black Bread Mold Overview published in their Agricultural Sciences database, and the Mold Busters Library's comprehensive Neurospora crassa pathogen profile updated in May 2022, alongside peer-reviewed entries from applied and environmental microbiology and studies in mycology's genus-level taxonomic revisions. Now back to the information. Vuillemine renamed it Rhizopus stolonifer in 1902. The name stuck because the fungus itself sticks everywhere. Tropical climates, subtropical zones, Arctic storage facilities. Wherever organic matter decays, Rhizopus moves in. It belongs to Zygomycota, a phylum unified by coenocytic hyphae, filaments packed with multiple nuclei and no cross walls. The fungus grows from the tip, extending threads that branch into 3 distinct structures. Stolons run horizontally, spreading the colony sideways. Rhizoids anchor below, embedding into substrate. Sporangiophores rise vertically, up to 2.5 millimeters tall and 20 micrometers wide, crowned with black sporangia that hold thousands of spores. When you see black dots on bread, you are looking at reproductive factories. Rhizopus thrives between 25 and 30 degrees Celsius. Its thermal death point sits at 60 degrees Celsius for 10 minutes. Below that threshold, the spores survive, waiting for conditions to shift. The fungus tolerates acidity down to pH 2.2 and alkalinity up to 9.6. It digests food externally, secreting enzymes into the environment, breaking down starches and sugars into absorbable compounds. Humans digest inside a tube. Rhizopus digests the world around it. This makes the mold a decomposer, critical for nutrient cycling in soil ecosystems, but catastrophic for stored grain. Between 1991 and 1996, Fusarium and Rhizopus species caused $3 billion in crop losses across United States wheat and barley harvests. Rhizopus stolonifer spreads through bread in circular patches, starting light, white or blue, then darkening as sporangia mature. The mold roots penetrate deep. Cutting off the visible section leaves hyphae intact, and the colony regrows. Rhizopus arizus, also called Rhizopus oryzae, generates cortisone, a steroid hormone, and ferments sugars into ethyl alcohol. In Asia, multiple Rhizopus species ferment soybeans into tempeh and rice into traditional alcoholic beverages. The same organism that ruins bread feeds millions. But in humans with weakened immune systems, Rhizopus becomes lethal. Zygomycosis, now called mucormycosis, occurs when spores enter the respiratory tract. The fungus invades blood vessels, cutting off circulation. Tissue dies, necrosis spreads. Mortality rates exceed 60% in rhinocerebral mucormycosis, the form that starts in the sinuses and moves to the brain. Diabetic ketoacidosis patients face the highest risk because Rhizopus produces ketoreductase, an enzyme that metabolizes ketone bodies, fueling fungal growth inside acidic blood. Smelling spoiled food delivers spores directly into nasal passages. Inhalation exposure is constant. Sporangia have spiny walls. Spores measure 7 to 8 micrometers in diameter. Globose, hyaline, gray, or brownish. Mucor plumbaeus populates soil, hay, stored seeds, dung, house dust, old carpets, ventilation ducts. Indoor air samples from homes in northern Norway during winter identified mucor frequently. Shanghai reported 20 cases of hypersensitivity pneumonitis in teachers exposed to mucor-contaminated sugarcane. Cork workers in occupational settings develop suberosis, a lung disease linked to mucor and esparto fibers. One fatal anaphylaxis case followed ingestion of mucor-contaminated pancake mix in 2001. Mucor, cercinoloids, Mucor irregularis, Mucor hyemalis cause mucormycosis in immunocompromised hosts. The infection spreads through blood vessels, identical to Rhizopus, tissue necrosis, organ failure, death. The difference between Mucor and Rhizopus sits at the microscopic level. Rhizopus has rhizoids, Mucor does not. Both kill. Cladosporium herbarum also survives cold storage, but Penicillium thrives there. The mold produces chains of conidia, spores formed at the tips of branching conidiophores. Under magnification, the structures resemble brushes, hence the name from Latin penicillus, a paintbrush. Penicillium species synthesize secondary metabolites. Some are medicinal. Penicillium chrysogenum, formerly called Penicillium notatum, produces penicillin, the antibiotic Alexander Fleming discovered in 1928 when a penicillium spore contaminated a Staphylococcus culture plate in his London laboratory. That accident saved millions, but not all penicillium metabolites heal. Penicillium expansum generates patulin, a mycotoxin that contaminates apples and apple products. Patulin disrupts a cellular metabolism, damages DNA, suppresses immune function. Regulatory agencies set limits below 50 micrograms per kilogram in juices. Penicillium verrucosum produces ochratoxin A, a nephrotoxic and carcinogenic compound found in cereals and coffee. The International Agency for Research on Cancer classifies ochratoxin A as a possible human carcinogen, group 2B. Penicillium contamination in bakeries occurs through flour dust. Mills grind wheat contaminated with fungal spores. The dust becomes airborne, spreading through ventilation systems, coating equipment, settling on cooling racks. Even after baking kills active mold, spores from the environment recontaminate bread during slicing and wrapping. Modified atmosphere packaging with 60% carbon dioxide and 40% nitrogen extends shelf life to 24 days at 20 degrees Celsius by suppressing mold germination. Mustard essential oil in volatile phase at 1 to 10 micrograms per milliliter inhibits Penicillium commune, Penicillium roqueforti, Aspergillus flavus, and Endomyces fibuliger, a yeast that causes chalky bread. Chemical preservatives like sodium propionate, potassium propionate, calcium propionate block penicillium growth by disrupting cell membrane integrity. Propylparaben, butylated hydroxyanisole, octyl gallate at 500 parts per million completely inhibit Penicillium corolophyllum, Penicillium verrucosum, Aspergillus ochraceus, Cladosporium herbarum. The war against penicillium is chemical and atmospheric. Aspergillus flavus shows yellow-green topside, gold or red-brown reverse. Aspergillus fumigatus displays blue, green, or gray coloration. The genus includes over 400 species. 16 are pathogenic to humans. Aspergillus niger infects third most often. Aspergillus flavus ranks second. Aspergillus fumigatus leads. These molds grow on grain, nuts, fruits, vegetables, soil, decaying vegetation. Aspergillus flavus and Aspergillus parasiticus produce aflatoxins, the most intensively researched mycotoxins on the planet. Aflatoxin B1, aflatoxin B2, aflatoxin G1, aflatoxin G2. The B types fluoresce blue under ultraviolet light. The G types glow green. Aspergillus flavus synthesizes B aflatoxins. Aspergillus parasiticus makes both B and G. Aflatoxin B1 is the most toxic, a potent hepatocarcinogen. The USDA calls aflatoxins the best-known mycotoxins in the world. Flour was baked into bread, causing alimentary toxic elukia with 60% mortality. But aflatoxin operates differently. It does not kill immediately, it accumulates. Chronic exposure at low doses induces liver cancer. The International Agency for Research on Cancer classifies aflatoxin B1 as Group 1, a known human carcinogen. Epidemiological studies in Southeast Asia and Sub-Saharan Africa link dietary aflatoxin intake to liver cancer incidence. Kenya in 2004 suffered an acute outbreak. 125 confirmed deaths. Contaminated maize. Aspergillus flavus grows in soil and invades crops during drought stress. High temperatures between 27 and 30 degrees Celsius, moisture content above 7%, and damaged seeds create optimal conditions. The fungus colonizes peanuts, maize, cottonseed, tree nuts, rice, wheat, sorghum, cassava, chili peppers, sesame seeds, sunflower seeds, coffee. Once established, it secretes aflatoxin into the seed. Animals fed contaminated feed pass aflatoxin transformation products into milk as aflatoxin M1 and M2. Humans consume the toxin through multiple pathways. Regulatory agencies set tolerance limits. The United States Food and Drug Administration permits 20 parts per billion aflatoxin in food for human consumption, higher levels in animal feed. This compound intercalates into DNA, causing mutations in the p53 tumor suppressor gene. A single nucleotide substitution, codon 249, guanine to thymine, appears repeatedly in liver cancer patients from high exposure regions. The p53 protein normally halts cell division when DNA damage occurs. Mutated p53 cannot. Cells proliferate without control. Tumors form. Children exposed during development suffer stunted growth and delayed cognitive maturation. Aflatoxin immunosuppresses, reducing white blood cell counts, increasing susceptibility to infection. No animal species is immune to acute aflatoxin toxicity. Adult humans tolerate low-level exposure better than children, but chronic intake over years or decades guarantees harm. The only defense is prevention, proper storage, moisture control. Rejection of moldy grain. Grain drying post-harvest prevents contamination. Aspergillus requires water activity above 0.9 to grow. Below that threshold, the mold cannot germinate. Oxalic acid accumulates in high concentrations, disrupting calcium metabolism in animals. Livestock ingesting Aspergillus, Niger-contaminated feed, develop calcium depletion, physiological abnormalities, sometimes death. Aspergillus niger also synthesizes citric acid, used commercially in food and beverage production. The mold ferments sugars into the acid at industrial scale, a process discovered in the early 20th century. Aspergillus niger is considered generally recognized as safe by regulatory agencies when used in controlled fermentation. But in the environment, the mold contaminates. Aspergillus niger causes black mold disease in onions, garlic, peanuts. Crown rot in peanuts under hot, humid conditions. The fungus invades wounds, producing enzymes that degrade cell walls. Infected tissue softens, blackens, exudes foul odor. In humans, Aspergillus niger causes aspergillosis, primarily in immunocompromised patients. Lung infections. Fungal balls form in pre-existing cavities. Allergic bronchopulmonary aspergillosis develops in asthma patients, triggering severe attacks. Hearing loss linked to Aspergillus niger ear infections. Automycosis. Nigeria reported multiple cases. The fungus colonizes the ear canal, producing black spores visible on examination. Treatment requires antifungal agents and mechanical debridement. It spoils meat in cold storage, black spots on refrigerated beef. The mold appears on damp walls, wallpaper, carpets, wood, textiles, heating and cooling systems, window panes. Outdoors, it populates dead plant material, soil, decaying vegetation. Cladosporium spores saturate the air during spring and fall. Peak concentrations reach 2,000 to 50,000 spores per cubic meter in summer. Indoor levels correlate with outdoor counts. When outdoor spore loads rise, indoor contamination follows. Cladosporium colonizes after primary molds like Penicillium, Aspergillus versicolor, Wollemia sebae establish. Secondary colonizer, it feeds on compounds the first wave produced. Shapes include ovoid, lemon, oblong, spherical. Only small spores, about 0.6% of airborne total, penetrate terminal bronchi and alveoli in human lungs. Larger spores lodge in upper airways. Cladosporium herbarum is the most allergenic species. Two major allergens identified, Cla H1 and Cla H2. Sensitization occurs through inhalation of dried mycelia and house dust or airborne spores. Symptoms include allergic rhinitis, asthma, rhinoconjunctivitis, allergic alveolitis. One skin test study showed 42% of children under age 4 reacted to cladosporium. Adults with asthma experience exacerbations during high spore seasons. Rare infections occur in immunocompromised individuals. Chromoblastomycosis, a chronic skin infection causing scaly papules, nodules, ulcers. Phaeohyphomycosis, infection of skin or internal organs by pigmented fungi. These conditions require systemic antifungal treatment. Cladosporium does not produce major mycotoxins, but it generates volatile organic compounds associated with musty odors. The smell signals contamination. Finding cladosporium on bread means the loaf was stored in high humidity, possibly refrigerated, allowing cycrotolerant molds to colonize. Fusarium arrives in pink and purple, white colonies at first. Cream, salmon, cinnamon, yellow, red, violet, pink, magenta, fluffy or cottony texture. The genus contains over 145 species. 14 are economically significant crop pathogens. Fusarium graminearum causes fusarium head blight in wheat and barley, also called scab. Fusarium sporotrichoides produces T2 toxin. Fusarium verticillioides and Fusarium proliferatum synthesize fumonisins. Fusarium culmorum generates deoxynivalenol. These mycotoxins belong to three main classes: trichothecenes, fumonisins, zearalenone. Trichothecenes inhibit protein synthesis. Type A includes T2 toxin, 14 times more toxic than deoxynivalenol, the most widespread type B trichothecene. T2 toxin suppresses immune function, damages bone marrow, causes aleukia, a reduction in white blood cells. Fumonisins disrupt sphingolipid metabolism, leading to liver and kidney damage. Zearalenone is an estrogenic mycotoxin, mimicking estrogen, causing reproductive defects in livestock. Embryo. Infection occurs from anthysis through soft dough stage of kernel development. Infected spikelets bleach prematurely, turning straw-colored while healthy spikelets stay green. Entire heads can die. During prolonged wetness, pink to salmon-orange spore masses appear at spikelet attachments. Severely infected kernels become lightweight, chalky pink to red, called scabby kernels or tombstones. These are blown out with chaff during harvest if the combine is not adjusted. Fusarium graminearum contaminated grain tests positive for deoxynivalenol, also known as vomitoxin because it induces vomiting in swine and other animals. Humans consuming flour from scabby wheat or moldy corn report nausea and headaches lasting 2 to 4 days. Regulatory agencies set tolerance levels at 1 part per million deoxynivalenol in grains for human use. Higher concentrations permitted in animal feed. Bread baked from contaminated wheat delivered the toxin at lethal doses. Symptoms began with abdominal pain, diarrhea, vomiting, prostration. Within days, fever, chills, myalgias, bone marrow depression with granulocytopenia, secondary sepsis. Further progression brought pharyngeal ulceration, diffuse bleeding into skin, petechiae, ecchymoses, melena, bloody diarrhea, hematuria, hematemesis, epistaxis, vaginal bleeding, pancytopenia, gastrointestinal ulceration. Mortality reached 60%. Research identified Fusarium sporotrichoides contamination in affected grain in 1932, spurring investigations for medical purposes and biological warfare applications. T2 toxin is one of the most potent natural toxins known. It weaponizes easily. The Soviet program stockpiled it. Whether it was deployed remains classified. Fusarium species contaminate bread post-baking. Flour dust carries spores. Bakery air circulates contamination. Slicing equipment transfers mold. Wrapping traps moisture. Fusarium grows on the surface, forming pink or purple colonies that deepen with age. The Observer from a ResearchGate publication in October 2019 documented bread mold growth for 8 days. By day 7, small whitish cream-colored colonies turned purple and dark peach. Approximately 80% of the slice covered in fungal growth. The slice appeared moist, darker. Those colonies were Fusarium species. Mycotoxin production occurred. Trichothecenes and fuminicins accumulating. Consumption would deliver toxins directly into the digestive tract. The bread becomes poison. Orange bread fungus, the only mold on this list that glows. French bakeries in 1843 first recorded the fungus forming reddish colonies on bread and pastries, named Penicillium psittophyllum by Montaigne, Oidium aurantiacum by Leveillé. Neither name held. The organism belongs to Ascomycota, not Penicillium. In 1848, Anselm Payen reported it resisted temperatures above 100 degrees Celsius, a fact that fueled spontaneous generation debates. The fungus survives heat that kills most molds. Neurospora psittophylla produces carotenoid pigments, specifically neurosporaxanthin, giving colonies orange or red color. When dark-grown cultures are exposed to light, they turn colorless initially, then pink or red within an hour, finally settling into orange. The ephemeral red stage is rarely seen. Most observers encounter orange. The fungus contaminates bakeries extensively before antifungal agents and improved hygiene. Yassine and Wheels in 1992 published a study in Journal of Applied Bacteriology documenting Neurospora species in bakeries. Multiple strains identified, 345 wild isolates collected from silage, Indonesian antjam, wheat grains, flour, bread, crumpets, bakery air, bakery dust. Neurospora crassa accounted for the bulk, conflicting with assumptions that Neurospora psittophylla dominates. Heterothallic strains of both mating types present. Variability indicated continual infection from a large population, estimated around 170 strains using Poisson distribution analysis. No known cases of home contamination or diseases caused by these fungi. Edward Tatum and George Wells Beadle used Neurospora crassa in experiments that won the Nobel Prize in Physiology or Medicine in 1958. They exposed the fungus to X-rays, inducing mutations, then observed metabolic pathway failures caused by errors in specific enzymes. This led to the one gene, one enzyme hypothesis, proposing that specific genes code for specific proteins. Beadle and Tatum founded biochemical genetics, which became molecular genetics. Neurospora crassa genome sequenced in 2003, 43 megabases long, approximately 10,000 genes. The fungus is a model organism for circadian rhythms, epigenetics, gene silencing, cell polarity, cell fusion, development. It grows fast, requires minimal nutrients, has a haploid life cycle simplifying genetic analysis. Despite its scientific importance, Neurospora remains a contaminant. It spoils bread, creates occupational asthma in wood and cork industries, triggers allergic reactions. Inhalation of spores causes sinusitis. Skin contact produces itching, rash, hives, eczema. Eye exposure leads to redness, itching, tearing, conjunctivitis. Nasal symptoms include congestion, runny nose. Systemic symptoms like headache, fatigue, fever occur rarely. The mold is not toxic. It produces no mycotoxins, but it colonizes aggressively. Neurospora psittophylla, linked to occupational asthma in 2010 study. Cork workers, wood processors, exposed to high spore loads develop respiratory issues over time. Single-celled fungus causing white, chalky discoloration. Hypopiccia bertoni, the updated name, contaminates bread during cooling and wrapping. Modified atmosphere packaging with mustard essential oil inhibits it at 1 to 10 micrograms per milliliter volatile phase. Chalky bread loses texture, becomes crumbly, unpalatable. Not toxic, just ruined. Flour dust introduced contamination pre-baking. Slicing equipment transferred colonies. Each species waits for its window. Rhizopus dominates room temperature storage. Penicillium takes refrigeration. Cladosporium follows cold. Fusarium thrives in moisture. Aspergillus colonizes anytime. Neurospora appears when heat-resistant spores survive. The competition between species determines which color you see. Rhizopus spreads fastest, occupying territory before others establish. Aspergillus holds ground in dry conditions. Penicillium wins cold wars. Fusarium claims wet environments. The bread becomes a petri dish. Sorbic acid, benzoic acid. Their salts create acidic conditions hostile to fungi. Ethanol sprays at 40 to 60% concentration kill spores on contact. Modified atmosphere packaging replaces oxygen with carbon dioxide and nitrogen, suffocating aerobic molds. Freezing halts growth but does not kill spores. Thawing reactivates them. Refrigeration slows reproduction, but cycrotolerant species like Penicillium and Cladosporium adapt. The only guaranteed prevention is consumption before contamination. Bread lasts 3 to 5 days at room temperature, 7 to 10 days refrigerated, 3 months frozen. Those windows close fast. Soft bread like white sandwich loaves allows rapid penetration. Dense breads like rye or sourdough slow it, but no bread is immune. The moment you see color, the entire loaf is compromised. Some species produce toxins that diffuse through the product. Aflatoxin in Aspergillus contaminated bread spreads beyond the visible colony. Deoxynivalenol in Fusarium infected grain permeates flour. Patalin from Penicillium expansum migrates through moisture. Mycotoxins are stable compounds. Heat does not destroy them. Baking bread from contaminated flour traps the toxins inside. They persist through cooking into consumption into your body. The dose determines harm. Low-level chronic exposure accumulates over years. Acute high-dose ingestion causes immediate illness. The line between safe and dangerous is invisible. Aflatoxin levels measured by high-performance liquid chromatography, enzyme-linked immunosorbent assay, thin-layer chromatography. Deoxynivalenol quantified similarly. Regulations enforce limits. The European Union permits 2 micrograms per kilogram aflatoxin B1 in cereals, 4 micrograms per kilogram total aflatoxins. The United States allows 20 parts per billion. Japan sets 10 parts per billion. Countries differ. The toxin does not. Grain elevators reject loads above threshold. Farmers lose income. Consumers buy contaminated product unknowingly when enforcement fails. Developing nations lack testing infrastructure. Contamination goes undetected. People eat poison daily. Liver cancer rates climb. The connection is established. Aflatoxin exposure correlates with hepatocellular carcinoma. The data is clear. Southeast Asia, Sub-Saharan Africa. Regions with high aflatoxin intake show elevated liver cancer incidence. The mycotoxin operates across decades. Childhood exposure predisposes adults. No amount is safe. Ergot poisoning from Claviceps purpurea on rye, not bread mold, but related, caused convulsions, hallucinations, gangrene. St. Anthony's fire. Mass outbreaks in medieval Europe. Entire villages afflicted. The connection between mold and disease went unrecognized. Folk remedies developed. Salt preservation, drying, smoking. These methods reduced water activity, preventing fungal growth. The science emerged later. Louis Pasteur in the 19th century proved microorganisms cause fermentation and disease. Robert Koch established postulates linking specific pathogens to specific illnesses. Mycology as a discipline followed. Taxonomy classified fungi. Aspergillus, Penicillium, Rhizopus, Mucor, Fusarium, Cladosporium, Neurospora. Each genus studied. Life cycles mapped. Toxins identified. Industrial uses discovered. Penicillin revolutionized medicine. Citric acid production transformed food manufacturing. Tempeh fermentation fed populations. The same organisms that destroy bread build civilizations. Bread mold performs this function on your counter. The loaf is dead plant tissue. Wheat seeds ground into flour, rehydrated, baked. The mold consumes what remains, returning carbon dioxide, water, simple molecules to the environment. From the fungus perspective, bread is a resource, a temporary abundance of sugar and starch. From your perspective, bread is food and mold is theft. The conflict is ecological. Two heterotrophs competing for the same substrate. You eat bread, mold eats bread. The faster you consume, the less mold gets. The faster mold colonizes, the less you get. Speed decides. Condensation forms, spores germinate. Hyphae extend, colonies establish. You open the bag, smell hits first. Musty, earthy, sour. Then you see color, green Penicillium, black Rhizopus, pink Fusarium, orange Neurospora. The diversity reflects your environment. Outdoor air contributes Cladosporium. Refrigeration selects Penicillium. Room temperature favors Rhizopus. Moisture invites Fusarium. High temperature tolerates Aspergillus. The mold profile is a fingerprint of your storage conditions. Antimicrobial surfaces. Calcium propionate added to dough at 0.2%. Sorbic acid sprayed on equipment. Staff trained in hygiene. Gloves changed hourly. Hairnets mandatory. The battle is constant. One lapse, one spore, one ideal condition, and contamination spreads. A single Rhizopus colony on one loaf releases millions of spores. The bakery becomes an incubator. Product recalls cost thousands. Reputational damage lasts longer. Food safety is not optional. The visible mold is the colony's reproductive phase. The invisible mycelium is the feeding phase. Already established, already metabolizing, already producing compounds. Some compounds are benign, some are deadly. You cannot tell by looking. Penicillium may be making penicillin or patulin or ochratoxin. Aspergillus may be synthesizing citric acid or aflatoxin. Fusarium may be producing industrial enzymes or trichothecenes. The mold does not broadcast its metabolite profile. You gamble every time you consume moldy food. Sometimes you win, sometimes you lose. The stakes are cancer, liver failure, neurological damage, death. Not immediately, not visibly. Years later, decades later, the mycotoxin accumulated. The DNA mutated. The cells divided uncontrolled. The tumor formed. The connection to that moldy bread you ate in your 20s is lost. The cause is invisible. The effect is fatal. See mold, throw it out. The financial loss of one loaf is negligible. The health cost of mycotoxin exposure is immeasurable. Longer shelf life means broader distribution, less waste, higher margins. Retailers demand extended dates. Consumers expect fresh taste days after purchase. Mold is the enemy. Billions spent fighting microscopic fungi. The fungi adapt. Resistance emerges. Penicillium strains tolerate higher propionate concentrations. Aspergillus species survive lower water activity. Fusarium colonizes despite modified atmosphere. Evolution operates on all scales. The molds that survive human intervention pass traits to offspring. Spore production rates increase, germination times shorten, enzyme efficiency improves. The arms race continues. Humans develop new preservatives. Molds develop new resistance. Neither side wins permanently. The balance shifts. Orange Neurospora, yellow Aspergillus, brown Cladosporium, each species a soldier, each colony an army. The objective is consumption. They consume bread. You consume bread. The competition is direct. Your advantage is speed and technology. Their advantage is numbers and persistence. Spores outnumber humans billions to one. Mold colonies outpace human action. The only guaranteed victory is elimination. Eat the bread before mold arrives. Destroy the bread once mold lands. There is no middle ground. Rotated storage. The mold persisted. Because it is everywhere. It does not invade. It is already there, waiting. Your bread left the bakery clean, the bag sealed. The spores inside came from your kitchen, from your hands, from the air you breathed into the bag when you reached in for a slice, from the counter you placed the loaf on, from the drawer you stored it in. The contamination is domestic. The source is environmental. The inevitability is absolute. Mold will grow. Time guarantees it. The question is when, not if. The answer depends on conditions. Warm, moist, exposed, fast, cold, dry, sealed. Slow, but never, never. If conditions are favorable, germination begins within 6 to 12 hours. The spore swells, the cell wall ruptures, a germ tube emerges, the first hypha. It extends at rates measured in micrometers per hour initially, accelerating as the mycelium establishes. Rhizopus stolonifer grows the fastest, covering a bread slice in 48 hours under optimal conditions. Aspergillus species take 72 to 96 hours. Penicillium requires 4 to 5 days. The variation reflects different strategies. Rhizopus is the sprint runner occupying territory before competition arrives. Aspergillus is the marathon runner tolerating harsh conditions, waiting out competitors. Penicillium is the cold weather specialist, thriving when others cannot. Proteases cleave proteins into amino acids. Lipases split fats into fatty acids and glycerol. Cellulases degrade cellulose in whole grain breads. The enzymes diffuse into the bread matrix, performing extracellular digestion, transforming complex molecules into absorbable units. The hyphae absorb the products, transporting them to growing regions, fueling expansion. This process generates heat. Large mold colonies raise local temperatures by 2 to 3 degrees Celsius. The warmth accelerates metabolism, speeding growth, creating a positive feedback loop. The hotter the colony, the faster it grows. The faster it grows, the hotter it becomes. Eventually, heat dissipation balances heat production, establishing equilibrium. But by then, the bread is lost. Aerobic fungi require oxygen for energy metabolism. They consume it from air trapped in bread's porous structure. As oxygen depletes, carbon dioxide accumulates. The gas builds pressure inside sealed bags, sometimes causing bags to inflate. You have seen this. The bag puffs. Inside, mold thrives. The carbon dioxide concentration reaches 5 to 10%, levels that would suppress some bacteria, but do not inhibit most molds. Aspergillus tolerates up to 15% carbon dioxide. Penicillium handles 20%. Only extreme modified atmosphere packaging with 60 to 80% carbon dioxide actually stops them. Home storage cannot achieve this. The bread breathes, the bag leaks, the mold wins. Bread typically measures 0.94 to 0.98, well above the 0.9 threshold that Aspergillus requires, far above the 0.85 that Rhizopus needs. Only xerophilic molds like Wallemia sebae and Erodium species grow below 0.8. Bread never reaches that dryness naturally. It would be hard as rock, inedible. The moisture bread needs for softness is the moisture mold needs for growth. The two requirements coincide perfectly. You cannot have soft bread without risking mold. Industrial solutions exist. Intermediate moisture foods, engineered to 0.75 to 0.85 water activity, resist mold while maintaining texture through humectants like glycerol and propylene glycol. But bread is traditional. Consumers expect it soft, moist, fresh. The market demands vulnerability. Spore concentration in your kitchen varies by season, geography, lifestyle. Summer outdoor air in temperate zones carries 50,000 to 100,000 fungal spores per cubic meter. Winter drops to 5,000 to 10,000. Indoor air typically contains 10 to 20% of outdoor levels when windows are closed, 50 to 70% when windows are open. Cooking increases humidity, raising spore germination rates. Houseplants contribute spores from soil fungi. Pets track in spores on fur and paws. Ventilation systems recirculate spores trapped in filters. Your body sheds skin cells constantly, each cell a potential substrate for mold. House dust contains fungal fragments, high-fall pieces, broken conidia. Vacuuming suspends them. They resettle everywhere. The bread on your counter sits in this environment. Thousands of spores land daily. Most do not germinate. Conditions are wrong. Surface is dry, temperature is low, nutrients are inaccessible, but one spore finds a crack. One droplet of condensation, one grain of flour dust, that is enough. Drought-stressed crops in sub-Saharan Africa and Southeast Asia suffer highest aflatoxin contamination. Aspergillus parasiticus appears in similar zones. Penicillium species proliferate in temperate climates. Europe and North America report high penicillium counts in indoor air. Cladosporium herbarum is cosmopolitan, but peaks in spring and fall in temperate regions. Fusarium graminearum thrives in regions with wet springs, infecting wheat during flowering when rain is frequent. Rhizopus stolonifer occurs globally, but concentrations are highest in humid tropics. Neurospora species are heat-resistant, appearing in warm climates and persisting where other molds cannot. Mucor species inhabit every environment but thrive in cold, damp conditions, growing in refrigerators where Rhizopus fails. The history of mold preservation spans millennia. Ancient Egyptians used honey, salt, and natron to prevent spoilage. Honey's low water activity, typically 0.5 to 0.6, inhibits all mold growth. Salt desiccates, pulling water from both bread and mold, halting germination. Natron, a mixture of sodium carbonate and sodium bicarbonate, raises pH above 9, creating alkaline conditions hostile to most fungi. Medieval Europeans smoked bread, coating it in phenolic compounds that disrupt fungal cell membranes. Vinegar pickling reduced pH below 4, acidifying substrates. Alcohol preservation, soaking bread in wine or spirits, denatured fungal proteins. These methods worked. Treated bread lasted weeks or months, but taste suffered. Modern consumers reject heavily salted, smoked, or pickled bread. Palatability trumps preservation. The industry seeks invisible solutions. Calcium propionate is tasteless at effective concentrations. Sorbic acid adds slight tanginess at high levels but remains acceptable. Natamycin, a fungal-derived antifungal, is odorless, colorless, flavorless, approved for surface application on cheese and cured meats. It binds ergosterol and fungal membranes, creating pores, killing cells. Natamycin sprayed on sliced bread extends shelf life by 7 to 10 days, but cost limits adoption. Preservatives remain the standard. Rejection rates for non-compliant shipments exceed 10% from high-risk countries. The United States relies on tolerance levels set by the Food and Drug Administration and action levels established by the United States Department of Agriculture. Testing is less comprehensive. Contaminated grain sometimes enters the food supply. Consumer advocacy groups push for stricter standards. Industry lobbies resist, citing costs. The balance shifts slowly. China implemented national standards for multiple mycotoxins in 2017, improving food safety but creating trade barriers. Exporters to China must demonstrate compliance through certified laboratory testing. Small-scale farmers cannot afford testing. They sell locally. Local consumers bear the risk. Mycotoxin exposure remains highest in low-income populations consuming untested grain. Mold identification requires expertise. Morphological examination under microscope reveals hyphal structure, spore shape, reproductive organs, but many Guernaria look similar at low magnification. Definitive identification demands spore measurements, culture on selective media, observation of colony growth patterns, sometimes DNA sequencing. Polymerase chain reaction amplifies specific gene sequences unique to each species. The internal transcribed spacer region of ribosomal DNA serves as a barcode. Sequence comparison to databases identifies the organism. Commercial testing labs offer this service. Cost is $50 to $150 per sample. Turnaround time is 3 to 7 days. Most consumers never test. They see mold, they discard bread. The species remains unknown. Whether it produced toxins stays uncertain. The risk is assumed acceptable. Usually it is. Occasionally it is not. The difference is invisible. UV light from sunshine provides additional sterilization. Wipe surfaces with alcohol at 70% concentration. Ethanol denatures proteins, disrupts membranes, prevents regrowth. Inspect adjacent items for contamination. Spores spread through contact. One moldy loaf contaminates the shelf. Check other bread, packaged foods, the bread box itself. Wash textiles that contacted mold in hot water at 60 degrees Celsius minimum. Heat kills spores. Cold washing preserves them. Air dry and sunlight when possible. UV radiation damages DNA, preventing germination. Professional remediation becomes necessary when contamination exceeds 10 square feet or affects porous materials like drywall. These situations require containment, negative air pressure, HEPA filtration, antimicrobial treatment, material removal. Cost runs thousands of dollars. Prevention costs nothing. The future of mold control involves genetic engineering. Researchers develop wheat varieties resistant to Fusarium head blight through insertion of antifungal peptide genes. Field trials show reduced infection rates, lower deoxynivalenol levels. Regulatory approval progresses slowly. Consumer acceptance lags. Genetically modified organisms face opposition despite safety data. Alternative approaches include biocontrol. Applying non-aflatoxigenic strains of Aspergillus flavus to fields outcompetes toxigenic strains, reducing aflatoxin in harvested grain. The method works. Several countries deploy it commercially. The non-toxigenic strain colonizes substrate, preventing toxigenic strain establishment. No genetic modification needed. Consumer acceptance is higher, but efficacy varies. Environmental factors influence competition. Drought stress, high temperature, insect damage create openings for toxigenic strains despite biocontrol presence. Titanium dioxide nanoparticles provide photocatalytic antimicrobial action. Under UV light, they generate reactive oxygen species that kill microbes. Zinc oxide nanoparticles function similarly. These materials integrate into plastic films, paper coatings, even dough formulations. Testing continues. Safety assessments are ongoing. Nanoparticle migration into food raises questions. Regulatory agencies require toxicity data before approving widespread use. The technology is promising, but not yet proven safe. Most exposure causes no lasting damage, but some mold is deadly. Some toxins are cumulative. Some effects are delayed. You cannot distinguish by looking. You cannot detect by tasting. Trust the timeline. Respect the science. When mold appears, the bread is gone, not just the visible section. The entire loaf. The mycelium runs through it. The metabolites permeate it. The contamination is complete. Throw it out. The cost is minimal. The safety is absolute. The decision is easy.